Go turtle.
This is herpes. The pathophysiology of the herpes simplex virus. So HSV spreads one of two ways, either skin-to-skin contact or sexual transmission. HSV1 is typically present above the waist, and HSV2 is typically present below the waist. Approximately 2/3 of the population is living with this cute little disease right here. It's super cute from the pictures.
Okay, the structure of the virus. The herpes simplex virus is a large enveloped virus that contains double-stranded DNA surrounded by an icosahedral capsid made of proteins which protects the virus's genetic structure. So this shape is similar to like a soccer ball. The capsid is then enclosed by an envelope of glycoproteins, which are like those cute little spikies that help the virus attach to the host. So how does HSV attack cells in our body? So first, the HSV infects our healthy cells. So that cute little green thing is the herpes virus, and it has its own viral DNA, that yellow, and proteins which helped it to infect the cells. The nucleocapsid outside helps the virus inject its DNA and proteins into the host cell. So once it's injected, the proteins help the DNA get into the host cell. The viral DNA and its proteins are now inside the cell, and it will incorporate itself with the human DNA and is now in the nucleus of the host cell. From here, it can do one of two things.
Now that the herpes is incorporated into the cell, the herpes virus is then mass produced. So first, the cell can mass produce, and then each replication of that reproduction will have its own copy of the viral DNA and proteins. After mass production is the lytic phase, in which the cell ruptures. So many viral nucleocapsids are produced that the cell can no longer support these viruses. So much pressure is placed on the cell membrane that it bursts or lyses, killing the cell. This is called the lytic phase. The virus leaves its host and infects other healthy cells, restarting the HSV life cycle all over again. Next is the lysogenic phase, or dormancy. The other step that may occur is the lysogenic phase. This is a phase of the virus's dormancy as it grows and continues to reproduce with its host cells. Because of this, people who have herpes have the virus for their whole lives. This virus may be dormant, however, it still has the potential to reach the lytic phase.
Fun fact, when herpes is in its dormant stage, excessive sunlight, excessive stress, and decreased immune system can trigger the virus to enter the lytic phase and the host cell bursts. This is super rockstar cool, as the herpes virus is able to read how the cell is responding to its environment.
So this diagram is a summarized representation of the HSV's life cycle. To reiterate what was explained before, these 5 events explain how HSV infects, lives in, and spreads to cells of the body.
Alrighty, the herpes simplex virus not the herpes, the herpes. The herpes simplex virus has a set of immune evasion genes that enhance its ability to replicate in the epithelial cells of your mouth. Next, I will briefly go over 3 of these gene products. First and foremost, ICP47. ICP47 is able to bind to and inhibit the human transporter of antigen presentation, which allows the virus to evade the host's immune response by suppressing antigen presentation to cytotoxic T lymphocytes. Next, glycoprotein E and glycoprotein I. These glycoproteins together compose the immunoglobulin Fc receptors of the herpes simplex virus, as you can see pictured here. Fc receptors, which are widespread among members of the herpes virus family, are proteins on the surface of immune system cells that bind to antibodies attached to pathogens. The herpes virus is able to use these Fc receptors to shield itself and other virus-infected cells from the host's immune response. Last but certainly not least, glycoproteins C1 and 2. These inhibit complement activation, which is a series of events in the immune system that complements the antiviral activity of antibodies. GC interacts with the C3b, which is a key enzyme of the complement pathway, by accelerating its decay. Hence, GC protects the herpes virus from complement-mediated neutralization. Done. Thank you for watching. Out.